Good morning. It's January 6th, and this is your daily brief in science. Here's everything you need to know. A Shanghai hospital team has announced a Phase 3 breakthrough in adjuvant therapy for early-stage triple-negative breast cancer, reporting strong reductions in recurrence and death. The multi-center study, led by Rujin Hospital, affiliated with Shanghai Jiaotong University School of Medicine, began in 2015 and culminated in results presented at an influential international conference. The trial evaluated adding carboplatin to the standard adjuvant chemotherapy regimen and included 786 patients across 19 hospitals in China. 3-year disease-free survival reached 93.1%, distant metastasis-free survival was 95.2%, and overall survival was 98.3%, indicating substantial benefit for operable patients. The China nationwide trial found post-operative recurrence reduced by 34% and mortality reduced by 61% with the carboplatin addition. Findings were presented at the San Antonio Breast Cancer Symposium, underscoring the study as a major advancement in adjuvant therapy for early-stage triple-negative breast cancer. Experts outside China welcomed the results, suggesting the therapy could influence global treatment guidelines with robust clinical data. Triple-negative breast cancer is notably resistant to treatment due to the lack of targeted therapies and high risk of recurrence and metastasis, making effective adjuvant options especially impactful. The multinational, multi-center collaboration highlighted international relevance and potential to reshape practice guidelines in global oncology. The therapy's reported outcomes position this approach as a potentially practice-changing option for operable patients with this aggressive cancer subtype. A new Nature Communications study shows how sunburn from ultraviolet exposure can undermine a key skin defense and raise cancer risk. Researchers found that ultraviolet light degrades the protective protein YTHDF2 in skin cells, lifting suppression on inflammatory responses that can drive tumor formation. A single allosteric site in the TRPM5 ion channel can both activate and inhibit the channel depending on the bound molecule, reframing TRPM5 regulation around a unified site with dual functionality. Led by Ruin and colleagues, the study identifies this site as a key regulator of activation, modulation, and inhibition within a calcium-permeable channel involved in taste, temperature, and pain sensing. TRPM5 sits among the TRP family and participates in sensory signaling, with implications for taste disorders and calcium signaling in broader systems. Cryo-EM analysis shows the site is strategically positioned and dynamically responsive to physiological cues, enabling the channel to adapt its behavior. Functional experiments demonstrate that allosteric modulators can enhance or suppress TRPM5 activity under different cellular conditions. This suggests potential for designing taste, modulating drugs, and treating related neurological conditions, while also informing how regulation might operate in other TRP channels. The findings point to a unifying regulatory concept where a single hub coordinates activation, modulation, and inhibition, challenging traditional bifurcated views of ion channel control. Researchers emphasize a multidisciplinary approach, combining computational modeling with experimental data to map this site and its effects under physiological contexts. The work opens avenues for future studies to reassess regulation across ion channels, map allosteric sites in the TRP family, and pursue targeted pharmacological interventions. Overall, the allosteric site functions as a multifunctional hub, supporting TRPM5 adaptability across diverse cellular contexts and signaling networks. Mitophagy is a central quality control process that selectively removes damaged mitochondria to maintain cellular homeostasis, with wide-reaching implications for health and disease. It acts as a master regulator of cellular health with therapeutic potential across neurodegenerative, metabolic, cardiovascular, immunological, oncological, and infectious diseases. New imaging and biomarker techniques now enable in vivo study of mitophagy with higher spatial and temporal resolution, supporting personalized medicine and assessment of therapeutic efficacy. In cancer, mitophagy plays a dual role. It can prevent tumor initiation by clearing dysfunctional mitochondria, but can also help established tumors survive metabolic stress and resist chemotherapy, necessitating nuanced strategies. Mitophagy influences metabolic diseases by shaping mitochondrial function in liver and adipose tissue. Impaired clearance promotes insulin resistance and inflammation, while enhancing mitophagy may improve metabolic parameters. Pathogens interact with mitophagy in diverse ways, informing antiviral and antibacterial strategies through host mitochondrial quality control dynamics. The pink 1/Parkin axis and alternative mitophagy signals coordinate tagging and degradation of damaged mitochondria, revealing multiple regulatory nodes for potential therapy. Dysregulated mitophagy is linked to neurodegenerative diseases like Parkinson's and Alzheimer's, contributing to neuronal loss, chronic inflammation, and cognitive decline, underscoring the need to restore mitophagic flux. Mitophagy intersects with immune regulation, shaping inflammatory responses and immune cell metabolism, offering avenues to treat autoimmune diseases and chronic inflammation. The overarching message? Understanding and precisely modulating mitophagy could transform disease management and promote health and longevity. Cardiovascular health hinges on mitochondrial quality control, with defective mitophagy driving hypertrophy, heart failure, and ischemic injury, pointing to cardiac protective mitophagy modulators. A long-term science effort aims to rewrite a living genome from scratch, assembling 16 Baker's yeast chromosomes in a coordinated, multi-institutional project. More than 200 researchers from over 10 institutions coordinated a redesigned genome built from 12 million base pairs, not by editing an existing genome, but by assembling chromosomes in large chunks and letting cells' own machinery integrate them. The team adopts a learning by building approach to gain insights into biology that affect environments, food systems, and medicines. They developed advanced debugging tools like pooled PCR tag mapping and CRISPR debugs to screen for and identify problematic genetic changes during the build. Researchers faced psychological and technical challenges, sustaining a long, slow progress project and maintaining cells that are difficult to grow. The team documented systematic bugs and avoided pitfalls, noting issues such as watermarks disrupting gene function and non-essential gene removals causing growth problems. Dr. Hugh Gould led the SynX6 chromosome work for the New South Wales Department of Primary Industries and Regional Development. The project also explores next-generation crops, including plans to build the world's first synthetic crop chromosome by constructing plant chromosomes inside yeast before transfer to plant cells. Macquarie University contributed to synthesizing 2 chromosomes, applying design principles like removing unstable elements, inserting watermarks, and enabling scramble for gene rearrangement testing. A Nature Biotechnology paper presents lessons from a decade-long effort to build the world's first synthetic eukaryotic genome, aiming to accelerate development of engineered organisms. Technical hurdles included yeast's inability to regenerate mitochondrial genomes, requiring genetic rescue and mitochondrial reintroduction via breeding. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.